Namaste to all. This is Dr. Srinath Naganathan, working as Assistant Professor in Biotechnology. In today's session, we are going to continue with the experiments to prove that DNA is the genetic material. As we have seen in the Griffiths experiment and in the Avery, MacLeod, and McCarthy experiment, that they proved from their experiments of transformation that DNA is the genetic material. So in this experiment by Hershey and Chase, they are actually confidently they proved it that the DNA is the genetic material by using another method of transformation using bacteriophages and bacteria. Let's see this experiment. So this experiment, they performed it in 1952. So in this, they have used the T2 bacteriophages and Escherichia coli for the experiment. So if you see the T2 bacteriophages, this is actually a model of T4 basically, but it is commonly the T2 bacteriophages which they have used. So the T2 bacteriophages, they have a hexagonal shaped head which is having the genetic material DNA. This hexagonal shaped material is made up of protein which is called as protein capsid. It is having a small collar. Beneath this, there is a spring-like protein sheath which is present. Below this, there is a base plate which is having a protrusion. And it is attached with tail regions like fibers which can hold on the surface of the E. coli or any bacteria. From this collar, from this, if they want to transfer their genetic material, they make an insertion like an injection on the bacteria and they penetrate through the cell wall and then they make their DNA materials to enter inside the organisms. Let's see how it does. So the pre-work, what they have done in this experiment is, Hershey and Chase, they actually cultivated the T2 phages and they have grown in two separate medias, which is containing one with S35, which is sulfur, S35 radioactive isotope, and the other one with P32 radioactive isotope. As you know, sulfur is a part of amino acids, cysteine and methionine. So they get incorporated in the amino acids of cysteine and methionine and becoming as protein during the translation process. Once the translation process is done, if you observe the T2 phages capsid, protein capsid, you can find it is having the protein S35. In the similar manner, phosphate is a part of the DNA backbone. So when this P32 radioactive isotope is incorporated in the medium, it gets incorporated in the DNA. And in the DNA of the bacteriophages, it will have the P32. So from this model, what we can understand is to know that whether the protein is getting transferred or the DNA is getting transferred. Okay, so for this, what we have to understand is how the bacteriophages, they actually transform this DNA to the E. coli. So the phage has four processes for this. The first process is landing. Once they get landed on the surface of the cell of surface of the organisms, they actually by pinning they absorb on the surface. This two process is actually involved for the adsorption process. After the adsorption is done, the collar region which is there, along with the spring-like mechanism, they make an insertion in the cell wall. So by this insertion, they actually inject the DNA material what they are having. So this process is actually called as injection. Once they are entered in this process, after the injection is done, once they enter into the process, they actually undergo or use the system or the cell's machineries like for the replication or for the transcription, and they undergo the replication process and the transcription process for producing various kinds or various copies of their own DNA material as well as the capsid and their structures. Once they get assembled inside the cell and the space of the cell is filled with the bacteriophages, they undergo the cell lysis, and the organism in which they are present, the cell will break open and all the bacteriophages are released. So this is a normal process of bacteriophages. So in this process, in this experiment, what Hershey and Chase did was they allowed the organism till the injection process. After that, what they have done is in the experiment, they took two different test tubes. In the test tubes, they filled it with the media which is having the T2 bacteriophage, which is having the S35 and E. coli. And the other tube, they use the T2, P32 and E. coli. Then what they did was, you can observe this by this imaginary diagram what I am drawing here, which is happening inside this test tube. So the E. coli is present. So once the bacteriophages lodge and absorb on the surface and they go for the injection, they release their materials. Okay, so the same process occurs in this part also. So this also occurs. So once they give some time, because they have observed that how much time it will take for the injection, then replication, all these things they have observed during those days. So after this, what they have found was, after some time they immediately went for the centrifugation. Once the centrifugation is done, the idea is, since the injection is done, it will be loose, so it can be removed and E. coli will be separate. So what happens is, their observation is, once the centrifugation is done, the supernatant will contain the phage capsids and the pellet will have the bacteria. So in this particular experiment, they observed the supernatant to know that whether it is having the protein which has got transferred, or it is the DNA which has got the transferred here. So what they have observed from the first experiment is, the radioactivity was observed in the supernatant. So in the supernatant, they found that the protein in T2, that is S32, S35 is present. So from this, they concluded that since the protein capsid is lighter and present in the supernatant, it proves and shows that the protein is not going inside the host cell. In the second model of experiment, what they have observed was, they observed both the supernatant and pellet and found that in the pellet, it is having the radioactivity. So from this, they concluded that the bacteriophage has transformed or transferred or injected their DNA inside the E. coli. That is why it is having the DNA which is P32 DNA of the T2 bacteriophages. From this, they observed that the cell, since it is heavier, that is why it is present in the pellet. And the T2 phages, since it has injected the genetic material, that is DNA, inside the E. coli, which has been grown in the P32 radioactive isotope, it shows the radioactivity. From this, they concluded that and proved that DNA is the genetic material. So from the three experiments, what the Griffith has done, it created the base for understanding that DNA is the genetic material. From this, Avery and McLeod and McCarty, they used the same experiment by using the enzymatic action and proved it that it is the DNA which is causing, which is transforming and causing the mice to lead to death because of pneumonia. And Hershey and Chase were using the T2 bacteriophages and grown in the radioactive isotope S35 and P32. It proved that the DNA which is incorporated with the P32 is getting transformed inside the E. coli, and this proved that DNA is the genetic material. I hope this gave you some idea and it was useful for you to understand as well as to use it for your syllabus as well as for your understanding. See you in the next session about RNA as the genetic material.